Spermatogenesis is the formation of sperm in the seminiferous tubules of the testes. Let's discuss the normal structure of the testes. Surrounding the testes, a tough fibrous outer covering is present that protects and provides support to the testes. Layer is called the tunica albuginea. The testes are divided into around 200 to 300 small compartments called testicular lobules. Each lobule contains 1 to 4 seminiferous tubules, where the process of spermatogenesis occurs. There are 3 types of cells present in the testicular lobule. The Spermatogonia and Sertoli cells are present inside the seminiferous tubule, while the Leydig cells is present between the 2 seminiferous tubules. The process of spermatogenesis occurs in these seminiferous tubules. First, let's discuss how at puberty the process of spermatogenesis actually begins. At puberty, spermatogenesis begins due to hormonal regulation by the hypothalamus and pituitary gland. At puberty, the hypothalamus releases gonadotropin-releasing hormone, GnRH, into the blood. This GnRH, through the blood, reaches the anterior pituitary gland and activates specialized cells called gonadotroph cells to release 2 types of hormones into the blood. One is follicle-stimulating hormone, FSH, and the second is luteinizing hormone, LH. The LH and FSH are released from the blood into the testes. Luteinizing hormone acts on Leydig cells, and the Leydig cells release the testosterone hormone, while follicle-stimulating hormone acts on Sertoli cells, and Sertoli cells release androgen-binding protein. Androgen-binding protein binds to testosterone, concentrating it in the seminiferous tubules. So, inside the tubule, the level of testosterone increases. This testosterone activates the Spermatogonia cells to form the sperm through the process of spermatogenesis. Remember that there are 2 types of cells, haploid cells and diploid cells. In a haploid cell, there are a total of 23 chromosomes, 1, 2, 3, up to 23, but each chromosome is single, not in. As each chromosome is single, not in pairs, haploid cells are represented by N. In a haploid cell, there are a total of 23 chromosomes. Examples of haploid cells are sperm in males and egg cells, oocytes in female. On the other hand, diploid cells also contain 23 types of chromosomes, but each chromosome is present in pairs, not in single form. As each chromosome is present in pairs, diploid cells are represented by 2N. And the total number of chromosomes is 46. Examples of diploid cells are all body cells except sperm and egg cells. Here, show haploid and diploid cells. In a haploid cell, the nucleus contains 23 chromosomes, and all are single. While in a diploid cell, the nucleus contains all 23 chromosomes in pairs, so there are a total of 46 chromosomes in the diploid cell. Another concept that is important is the number of DNA strands in a chromosome. Normally, a chromosome is made up of DNA strands and histone proteins. The DNA strands are represented by C. In a haploid cell, each chromosome is single, so there is 1 DNA strand in each chromosome. Therefore, a haploid cell is represented by N and C. In a diploid cell, each chromosome is present in pairs, so there are 2 DNA strands. It is represented by 2N and 2C. Remember, if a chromosome is made up of 2 DNA strands as shown here, it is still considered 1 chromosome because chromosome numbers are counted based on the centromere. If there is a single centromere, it counts as 1 chromosome. In this image, there are 2 chromosomes because of the 2 centromeres. So the first one is represented by N because chromosome is single not in pair, and as there is 2 DNA strand in each chromosome, so it is represented by 2C. In the second one, as each chromosome is in pairs and contain 2 DNA, so represented by 2N and 2. In this last image, as each chromosome is in pair not single, so it is represented by 2N, and as there are total 4 DNA strands, so it is represented by 4. Now let's discuss the process of spermatogenesis. We already discussed that at puberty, the hypothalamus releases GnRH, gonadotropin-releasing hormone. This hormone acts on the anterior pituitary gland, which releases 2 hormones, FSH, follicle-stimulating hormone, and LH, luteinizing hormone. Luteinizing hormone, LH, acts on Leydig cells, stimulating them to release the hormone testosterone. Testosterone then acts on Spermatogonia cells and, through mitosis, divides them into 2 daughter cells, type A and type B cells. Type A cells continue to divide and produce more spermatogonia to maintain their population. Type B cells divide by mitosis into primary spermatocytes. Spermatogonia are diploid cells, meaning each chromosome is in pairs, 2N and 2C. Similarly, both type A and type B cells are also diploid, 2N and 2C. When primary spermatocytes arise from type B cells, they are also diploid, 2N and 2C. After this, primary spermatocytes replicate their DNA. Now, each chromosome contains 2 DNA strands, but it is still a diploid cell because each chromosome is still in pairs. At this point, the primary spermatocyte is represented by 2N and 4C. Primary spermatocytes then undergo meiosis I and divide into 2 secondary spermatocytes, with each cell receiving 23 chromosomes from the primary spermatocyte. These are haploid cells because they now contain only one set of chromosomes, though each chromosome still consists of 2 DNA strands, represented by N and 2C. Each secondary spermatocyte then undergoes meiosis II and divides into 2 roughly spherical cells called spermatids. These are also haploid, N, and each chromosome is made up of a single DNA strand, so they are represented by C. Each spermatogonium ultimately produces 4 spherical cells, known as spermatids. Through the process of spermiogenesis, each spermatid transforms into a mature motile sperm containing a head, neck, body, and tail. Spermiogenesis is the final stage of spermatogenesis, in which round immature spermatids are transformed into mature motile sperm consisting of a head, neck, body, and tail. First, the Golgi apparatus in the spermatid produces a vesicle called the acrosome and applies it to one pole of the nucleus in the form of a cap. This acrosome contains hydrolytic enzymes. These enzymes help break down the protective layer surrounding the egg, particularly the zona pellucida, a glycoprotein layer surrounding the egg, allowing the sperm to reach and fertilize the egg. The nucleus and the overlying acrosomal cap migrate to a position close to the cell membrane. This area, occupied by the nucleus and acrosomal cap, becomes the head of the developing sperm. As the acrosome is forming, the 2 centrioles move to the caudal pole of the cell, and one of them, the distal centriole, gives rise to a flagellum, which forms the tail of the sperm. The mitochondria partially fuse with one another and form a sheath around the proximal part of the flagellum. This area, containing the mitochondrial sheath, forms the body of the sperm. In the final stage of maturation, the residual cytoplasm is removed from spermatids in the form of residual bodies, which are phagocytosed by the Sertoli cells. Remember, a special feature of spermatogenesis is that with each successive division, the cells remain attached to one another through intercellular bridges up to the spermatid stage. They are only separated from each other when sperm is formed. The whole process of spermatogenesis occurs in the grooves formed by 2 Sertoli cells. After sperm is formed, it is released from the groove into the seminiferous tubule lumen, a process called spermiation. The sperm are still non-motile. This sperm from the lumen of the seminiferous tubule moves to the epididymis. Here in the epididymis, sperm mature and gain motility and are stored until ejaculation.